కంటే వేగవంతమైనది మనిషి ఆలోచనలు. అయితే మన ఆలోచనలకు అందని స్థాయిలో అనునిత్యం కొత్త పుంతలు తొక్కుతున్న టెక్నాలజీని కాలానుగుణంగా అనుసరించే వారి భవిష్యత్తు మాత్రం ఉజ్వలం అని అనడంలో సందేహం లేదు. నేటి మరియు రేపటి టెక్నాలజీ అంతా ఆర్టిఫిషియల్ ఇంటెలిజెన్స్ అండ్ మెషిన్ లెర్నింగ్ మరియు డేటా సైన్స్ మీదే ఆధారపడి నడుస్తుంది అని ఎంతమందికి తెలుసు? చేబ్రోల్ ఇంజనీరింగ్ కాలేజీలో 2020-21 విద్యా సంవత్సరం నుండి లేటెస్ట్ టెక్నాలజీకి అనుగుణంగా బీటెక్ లో కొత్త కోర్సుని చదువుకునే ఓ అద్భుత అవకాశం. నాక్ అక్రిడేషన్ మరియు 5 పేటెంట్స్ కలిగిన చేబ్రోల్ ఇంజనీరింగ్ కాలేజీలో కంప్యూటర్ సైన్స్ విభాగంలో ఆర్టిఫిషియల్ ఇంటెలిజెన్స్ అండ్ మెషిన్ లెర్నింగ్ లో 120 సీట్లు మరియు డేటా సైన్స్ లో 60 సీట్లు కలవు. కౌన్సిలింగ్ కోడ్ CHBR. నాణ్యమైన ఇంజనీరింగ్ విద్యతో పాటు 100% ఉద్యోగ అవకాశాలు కల్పిస్తున్న ఏకైక విద్యా సంస్థ చేబ్రోల్ ఇంజనీరింగ్ కాలేజ్, చేబ్రోలు, గుంటూరు, ఆంధ్రప్రదేశ్.
JNTUK, third year ECE, Clinical Laboratory Instruments, Biomedical Engineering, Friday, June 25. So clinical laboratory instruments. So everybody in our hospital environment, we have regular hospitality domain. We are familiar about laboratory instruments. For example, pathology department. So in pathology department, we have various clinical laboratory instruments like blood test, urine test, likewise pathology department. So out of them, here we have to know somewhat of the clinical instruments. So patient specimen is done in the clinical laboratory. So it provides information to the aid in the diagnosis of disease and evaluate the effectiveness of the therapy. So it provides information to aid in the diagnosis of disease and evaluate the effectiveness of the therapy. So the clinical laboratory, also known as clinical pathology department, for example, chemistry, hematology, microbiology, so all are comes under clinical laboratory criteria. So in this clinical laboratory, blood, urine, serum, and other fluids which are analyzed in the chemistry section to find the content of various clinical important substances. And the determination of the numbers and the characteristics of the elements in the blood that is red blood cells and white blood cells and platelets, and also functions of physiological systems in the blood, and that is blood clotting, etc., which are done in the hematology section. So the microbiology section helps in the studies on various body tissues and fluids to determine whether pathological microorganisms are existing in the particular theory is or not. So in all above electronic automation, which are quite common, so the mainframe and the microcomputers are keep track in the salient parameters. So the fast response and accuracy and precision are essential requirements for these clinical laboratory instruments. So let us look one of the clinical laboratory equipment, which is called a spectrophotometer. So this is the spectrophotometer. So this is the block diagram for spectrometer. So the main intention for this spectrometer is it is based upon the fact that substances of clinical intersect selectivity, which can absorb or emit the electromagnetic energy of different wavelengths, that is ultraviolet and visible or near the infrared. Most of the instruments operate in the visible range. The following diagram shows spectrophotometer, so which is having photometer and calorimeter individual operation. So the again subsystems which are having in the spectrophotometer power source, so which is a light intensity source. So this power source has a hydrogen discharge lamp for 200 or 36 nanometer range, and tungsten filament lamp for 360 to sub 800 nanometer range, and the wavelength selector. So this wavelength selector is glass filter and interference filter, so, and monochrome using prisms and different gratings. So wavelength selector, and which is having covered, so which you can hold substances being analyzed. 3 is the detector and readout. So this is called photometric system. Clear?
Effect of a sample on a beam of light. You can learn a lot about a substance by studying the way it interacts with light. Here's how a spectrophotometer works. A lamp provides the source of light. The beam of light strikes the diffraction grating, which works like a prism and separates the light into its component wavelengths. The grating is rotated so that only a specific wavelength of light reaches the exit slit. Then the light interacts with the sample. From this point, the detector measures the transmittance and absorbance of the sample. Transmittance refers to the amount of light that passes completely through the sample and strikes the detector. Absorbance is a measurement of light that is absorbed by the sample. The detector senses the light being transmitted through the sample and converts this information into a digital display. Let's practice by performing a simple experiment. In this exercise, we'll determine the absorbance spectrum of red food coloring. To begin, set the wavelength to 380 nanometers. With the sample chamber empty and the lid closed, set the display to 0% transmittance by using the zero control knob. There are two cuvettes. One contains only milli-Q water. This is our blank. Wipe the blank cuvette with a Kim wipe to remove any fingerprints, and place the cuvette into the sample chamber by aligning the guide marks. Adjust the display to 100% transmittance. Remove the cuvette with the blank solution. Now set the display mode to absorbance by pressing the mode control key. The second cuvette contains milli-Q water and red food coloring. This is our sample. The absorbance of the sample is 0.845. Press the mode control key to select transmittance. The transmittance for the sample is 14.2%. Increase the wavelength by 25 nanometers to 405. Wipe the blank cuvette again and place it in the sample chamber. Adjust the transmittance control knob until it displays 100%. Now remove the blank. Wipe the sample cuvette and place it in the sample chamber. The transmittance for the sample at 405 nanometers is 7.4%. The absorbance for the sample is 1.130. Record this in your lab notebook. Increase the wavelength by 25 nanometers to 430. Repeat these steps with the blank and sample, writing each value in your lab notebook. Increase the wavelength by another 25 nanometers to 455 and test the sample again.
Amount of light observed by the sample. So it measures the amount of light observed from the sample. The principle of spectrophotometer. Spectrophotometer techniques are mostly used to measure the concentration of solutes in solution. Solution is basically our test solution. By measuring the amount of light that is observed by the solution. Test solution. in a cuvette placed in the spectrophotometer. So it measures spectrophotometer measures the amount of light observed from the sample. The ion concentration or concentration of solute is directly proportional to the amount of light observed from the sample. So as their concentration or solutes are high, the absorption is also high. So in this way we measure our test sample. The working of spectrophotometer. The light source, monochromator, our sample, detector and digital display are used in spectrophotometer. First of all, a light source is used. The light source typically yields high output of polychromatic light. Then this polychromatic light is transferred to the lens. Lens collect all polychromatic light coming from the light source. So see, it collects all the light source. And then monochromator. Monochromator separate light into different component, means it accepts polychromatic input light and outputs monochromatic light. So now you see only one wavelength of light and it transmits from the our sample solution. Any photosensitive device can be used as a detector of radiant energy. The photocell and phototube are the simplest photodetector, producing current proportional to the intensity of light. Then display devices. The data from the detector are displayed by the readout devices such as analog, digital display, LCD, or output can also be transmitted to the computer or a printer. So from here we see our result, that how much light is observed from the sample or how much solutes are present in our test sample. Here is the application of spectrophotometer, and it is used in clinical chemistry, molecular biology, immunology, and hematology.
In this way, spectrophotometer will collects the sample and automatically the detector reading will able to provide the accuracy. Another spectrometer, another clinical laboratory equipment here. So block diagram for the gas liquid chromatometer. So gas and liquid chromatograph or GLC. So the block diagram has carrier gas supply, pressure control, injector, column, detector, recorder, and sample. So initially, carrier gas supply is able to produce the pressure to the pressure controller input. So pressure controller input regularly monitors the concerned pressure. So when pressure control output applied to injector, so we have already one sample as input signal. So from the input signal and the pressure control another input, injector monitor the column matrix as well as the detector. So three sections are called as here temperature control device. So from where the concerned output is recorded or plotted at recorder side. So this is the graph GLC recording for analysis of the blood. Figure A, that is a pKa equal to nanobacteria, and peak B corresponds to Heptabacterium, and peak C pinotin. So these are the three levels of individual blood sample information. So this is the another spectrometer, which is the same spectrometer, somewhat of modified model prototype. Same principle, mirror, lens, and mirror, lens, split filter, photo detector, etc. So another important spectrometer here, electro process. So nothing but by using this spectrometer, proteins in the plasma and blood and urine, which are identified by the antibodies, electro process in the moment of solid phase with respect to liquid, that is but buffer solution. So the mobility of a particle is directly related to net magnitude of the particle, which is having a high charge intensity. The temperature mobility is directly related to the temperature, so it will cause the medium temperature to increase. This decrease, which can decrease its resistance by increasing rate of migration. So the evaporation helps in the increase of this concentration of the particles. So the distance of the migratron is directly related to the time of electroprocess. Similarly, hematology. So by using the hematology, these are the devices which can measure characteristic of element, red blood cells and white blood cells and platelets, which is simply blood study. Chymography is another important bioclinical lab instrument. Chymograph. It is an instrument for continuous recording of heartbeats, muscle contraction, respiratory events, and it is comprised of a metal drum, a rotating spinal, and electronic motor. Another clinical instrument, galvic skin resistance, the GSR, galvic skin resistance. So it is the change in the skin resistance due to sweating. So the emotional state such as fear, panic, or alertness indication can be achieved through this GSR. An active electrode is fixed on the palm of the hand, and the second neutral electrode is placed on the wrist or back of the hand. So constant current is about 10 milliamperes is passed through the electrodes, and the GSR are be measured. So GSR is in the range approximately 500 kilowatts. So this is the way to measure the monochrome. So these are the information about the clinical lab equipment or instrument.